We are Group 8, and this is our CLC project.
Okay, so we're going to start with mechanisms of antiviral medication. Antiviral medications are both preventative and protective. They will help fight off viral infections as well as working to stop you from getting infected to begin with. Healthcare providers will typically only prescribe antiviral medications for conditions that are chronic or life-threatening. Most other viral infections will clear up on their own. Here are some examples of viruses for which doctors would administer drugs, as well as the type of drug prescribed. COVID-19, Ebola, flu, herpes, hepatitis B and C, HIV, and Mpox are all different types of viruses that doctors would prescribe antiviral medications for. And you guys can read all the different types of drugs that they would use. Antiviral medications work by blocking the attachment of viruses to the host cells, boosting the immune system, indirectly fighting viruses by enhancing the body's natural defenses, working to lower the amount of active viruses in the body, and they do this by inhibiting the replication of enzymes viruses use to copy their genetic material, such as DNA or RNA, and they also block enzymes like protease that cut viruses into functional pieces, and then finally they prevent the release of new viruses from infected cells. Okay, so lastly we have some examples of some specific types of drugs and how they work against specific viruses. So you have oseltamivir, which is normally called Tamiflu, and it's a drug that inhibits neuraminidase, which keeps flu viruses from leaving the infected host cell. And then you have tenofovir and entecavir, which are drugs that block viral DNA polymerase to stop the replication of hepatitis B. And then to prevent HIV, we use reverse transcriptase inhibitors like zidovudine and tenofovir.
Antimicrobial susceptibility testing, or AST for short, is a laboratory process used to determine which antibiotics can effectively inhibit or kill specific microorganisms. To perform this test, a bacterial sample is first isolated from the patient. The sample is then exposed to different antibiotics using methods like disk diffusion, broth dilution, or automated systems. After incubation, we measure either the zones of inhibition or the minimum inhibitory concentration, also known as the MIC, to see how well each antibiotic works. The main purpose of AST is to guide physicians in selecting the most effective antibiotic for treatment. This helps ensure patients receive the right therapy, prevents the misuse of antibiotics, and plays an important role in combating antimicrobial resistance. AST matters because it ensures targeted therapy which reduces side effects and the risk of resistance. It also protects public health by tracking resistant bacterial strains and supports infection control in hospitals and communities. On a global scale, AST helps address the growing threat of superbugs and promotes responsible antibiotic use, preserving their effectiveness for future generations. In the end, AST reflects both scientific precision and moral responsibility. It reminds us to test first, treat wisely, and care for life responsibly.
Talking about the mechanisms of resistance to antimicrobial drugs. The four main mechanisms are going to be limiting drug uptake, modification of the drug target, inactivation of the drug, and active efflux of a drug. So starting with modification of drug targets, going back to the gram-positive and gram-negative bacteria that we learned about earlier. In gram-positive bacteria, we see alterations in the structure or number of penicillin-binding proteins, and these alterations can decrease the amount of penicillin that binds to the target cell, or just inhibit the ability of the penicillin to bind to the target cell in general. And then in gram-negative bacteria, we see in antimicrobials like glycopeptides, such as vancomycin. Vancomycin inhibits cell wall synthesis. So in vancomycin resistance, like in VRE and MRSA, we have acquired genes that decrease the binding ability of vancomycin. And then a similar mechanism, some bacteria have developed resistance to lipopeptides. Lipopeptides polarize the cell membrane, and these mechanisms involve gene mutations that alter the charge of the cell wall and prevent the binding of certain mediators that are required for the antimicrobial to bind. This can be like calcium. Other modifications that can be seen, we have mutations in the ribosomes, DNA gyrase modifications, enzyme mutations. Moving on to limiting drug uptake. So in gram-negative bacteria, we see the bacterial cell wall coming back into play. And then in gram-negative bacteria, the lipopolysaccharide layer gives them resistance to large antimicrobial agents like penicillin. And then antimicrobial agents often enter gram-negative bacteria through cell porins, and the bacteria may decrease the number of porins in the outer membrane or increase the selectivity of the porin channels. And this often happens in transport channels. And then in gram-positive bacteria, we see that since gram-positive bacteria lack an outer membrane, some have started to produce a thickened cell wall to develop antimicrobial resistance. For example, Staphylococcus aureus has recently developed vancomycin resistance by this mechanism. And then lastly, for limiting drug uptake, we see biofilms. Biofilms are composed of a matrix of polysaccharides, proteins, and DNA that it gets from the bacteria that it houses. It makes it more difficult for antimicrobials and immune responses to reach the bacteria. And since bacteria biofilms tend to grow slowly, it makes them more resistant to antimicrobials that target growing cells like penicillin. And also, since bacteria and biofilms are in such close proximity, this may enable the sharing of antimicrobial resistance genes by means of horizontal gene transfer, since a requirement of that gene transfer was for them to be close to each other. And then lastly, we have drug inactivation and drug efflux. So drug inactivation happens by two mechanisms, either degradation of the drug, which is primarily seen in B-lactamases, which are enzymes that hydrolyze the drugs, or we see a transfer of a chemical group, mostly acetyl, phosphoryl, and adenyl groups. And then lastly, drug efflux. Bacterial cells contain efflux pumps that clean toxic substances, in this case antimicrobial drugs, out of the cell. And then the types of efflux channels are seen in this picture on the side. Pumps are activated or suppressed depending on environmental factors and available substrates that can bind to these enzymes. And then the resistance capability of the pumps depends on the carbon source available to the cell.
Hi everyone, I'm Justine Campos. I'll be talking about epidemiology, reservoirs of infection, and disease transmission. So what exactly is epidemiology? Epidemiology is the study of the distribution and causes of disease in populations, and this is important because its purpose is to identify the cause, similar patterns, along with methods to avert and administer the spread of diseases. Ignaz Semmelweis, also known as the father of hand hygiene, noticed that puerperal fever was drastically higher in women in a maternity hospital that he was working at in Vienna. He noticed that the women who were being treated by doctors who also happened to do autopsies had a higher incidence of 18%, which was 4 times higher than a regular section ran by midwives who did not perform any autopsies. He hypothesized that this poison was transferred from cadavers via unwashed hands, so he implemented hand washing with disinfectants. This poison was most likely Streptococcus pyogenes, and the fever incidence dropped drastically, approximately by 1/3 after everyone started implementing their hand washing with disinfectants. In epidemiology, there are communicable and non-communicable diseases. So infectious diseases that can be transmitted from one host to another, such as measles, colds, influenza, and even COVID-19, are known as communicable diseases. Non-communicable diseases are diseases that do not transfer from one host to another. This means that their microorganism causing the disease arose from an individual's normal microbiota. Leading into reservoirs of infection, these are the natural habitats of a pathogen, some of the potential sources of an infectious agent. These reservoirs of infections are important because they affect the range and distribution of a disease, and once identified, people who are susceptible can be inhibited from coming across or in contact with the source of the disease.
So there are three main types of reservoirs, starting off with the first one being human reservoirs. These are the most significant reservoirs for the majority of communicable diseases that were mentioned previously. Humans are the only reservoir which allows the disease to be easier to control because of the setup for prevention and control programs. It's simpler than it would be in wild animals, which leads me to the next one, which is non-human animal reservoirs. These are the source of many pathogens and can be very difficult to control, specifically in the wild animal communities, as I mentioned. So rats, rock squirrels, prairie dogs are the reservoirs for Yersinia pestis, which is the bacterium that causes the plague. So diseases such as rabies and the plague that can be transmitted to humans but exist primarily in other animals are called zoonoses. Zoonotic diseases are more commonly severe in humans than they would be in animal hosts due to them being accidental in humans. The last reservoir would be environmental reservoirs. These have few pathogens in their reservoirs, and the pathogens that have environmental reservoirs are difficult or impossible to eliminate. Clostridium botulinum obviously leads to botulism, and Clostridium tetani leads to tetanus, which are both widespread in soil. Ending with disease transmission. Disease transmission is how pathogens are transmitted from its reservoirs to the host. So there are two types of transmissions, direct and indirect transmission. Direct transmission involves the immediate transfer of an infectious agent from a reservoir to the appropriate host, which can be transferred via direct physical contact or projection of respiratory droplets onto mucous membranes. So physical contact deals with something as simple as a handshake or something as intimate as sexual intercourse. Projection of respiratory droplets could be inhaled of large microbes, laughing, sneezing, talking, or even coughing. Indirect transmission is the transfer of a pathogen from a reservoir to a host via airborne particles, vehicles, or vectors. Airborne transmission happens through inhaling microbe-containing airborne particles that may be suspended in the air due to their small size. So vehicle-borne transmissions are transmitted via fomites, foods, and water. Vector-borne transmissions are transmitted through any living organism that can transmit a disease causing a microbe like disease, flies, ticks, lice, and fleas. So an example of a mechanical vector would be a fly landing on feces and then going ahead and landing on food that someone will eat. And then biological vectors such as mosquitoes sucking a human's blood is a way that indirect transmission can be transferred.
Healthcare associated infections. These are defined to be infections that people get while receiving health care for another condition. These are also known as nosocomial infections. These can happen in hospitals, surgical centers, long-term care facilities, and in some sources of infection would include pathogens such as bacteria, fungi, or viruses, as well as contaminated equipment, healthcare personnel, invasive procedures, and endogenous flora. Some examples of healthcare-associated infections would include hospital-acquired pneumonia, which is often ventilator-associated, gastrointestinal infections, primary bloodstream infections, urinary tract infections, which are often due to the use of catheters within the hospital, lower respiratory tract infections, infections of the eye, ear, nose, and throat. Hospital-associated infections are considered to be major health concern because they increase patient illness and death and also may lead to longer hospital stays. Some of the causative agents of hospital-associated infections would include bacteria. Bacteria can arise from exogenous or endogenous sources, which means from inside the patient's body or from external things in the environment. An example of a bacteria that might be acquired in the hospital would be C. difficile, which is accounted for 15% of all healthcare associated infections, meaning that this is the most common bacterial infection. And this is multi-drug resistant bacteria, which is hard to treat.
Another one would be fungi. Fungi is mainly seen and spread through the uses of central lines and urinary catheters. The most common fungi healthcare associated infection would be C. albicans. Another one would be viruses. Viruses are the least common out of all three of these, as they account for 1 to 5% of all hospital associated infections. Some examples of this would include hepatitis B, hepatitis C, and are often associated with the unsafe needle practices within the hospital. Some risk factors of healthcare associated infections would include weakened immune system, catheters, IV lines, ventilators, and surgical procedures.
Okay, so for Christian worldview, mechanisms of antiviral medications can reflect this because in the Bible, Jesus tells us to help take care of the sick and vulnerable, specifically in Matthew 25:36, when he talks about how the disciples helped him when he was sick and they were called righteous for it. Acts of mercy are a reflection of serving God as well as stewardship and compassion. In 1 Peter 4:10, God tells us to use our spiritual and physical gifts wisely and to use them to help serve others. Antimicrobial susceptibility testing aligns with this principle because it uses testing to help find the most effective treatment to serve others. Mechanisms of resistance to antimicrobial drugs can relate to both of these principles we just talked about, because we can use this as a gift from God to help serve others as well as taking care of the sick and vulnerable. This topic can be seen as a metaphor in the Christian worldview for the struggle against sin. Resistance to drugs as well as God's will are natural processes that can be intensified with misuse. These can be addressed by emphasizing stewardship and the need for action and application of these tools. For epidemiology, understanding how the spread of diseases work can help us prevent harm and promote flourishing for others. This aligns with the Christian call to love and serve others. In Mark 12:31, God tells us to love our neighbor as ourselves. For healthcare associated infections, Christians are called to help take care of the sick and vulnerable, and healthcare professionals can do this by working to prevent infections and reducing avoidable suffering. And this goes back to Matthew 25:36, when we are called to help take care of the sick and vulnerable.